In this lecture, we're going to discuss blood type. Before I go to the blood type itself, I'm going to explain very briefly what is antigen, what is antibody, and how your immune system works. So that's where you have a clear understanding before we discuss the blood typing. There are different types of glycoproteins located on the surface of the surface of the cell, and those proteins are called antigens. There are about 50 different kinds of proteins located on the surface of the red blood cells. So we could say that there are 50 different antigens on the surface of the red blood cells. But 3 of those proteins are important, and those are protein antigen A, antigen B, and antigen D that we're going to discuss in a second. So what is an antigen? Antigen is basically a complex membrane glycoprotein that's located on the surface, and you could just, every single every cell has their unique shape and unique surface proteins, and your immune system recognizes those proteins as self and doesn't attack them. And any protein that's not recognized as self, they will be attacked. So let's look at the cell, for instance. If you look at this cell over here, this cell has protein, and the protein shape is like this, and we call this antigen. This cell has a different protein, and this protein has this shape, we call it antigen. And this cell has this shape protein. This cell has over here this shape protein. So as you could see, every single cell has their own shapes. So your immune system recognizes the proteins that are located on the surface of your own body cells as self. Any of those proteins that's not recognized by your immune system as self, for instance, like macrophages will go around and look at those, and if they don't recognize those cells, they will stimulate the immune system and produce antibodies against it. Now, you could think of that shape that you see over here, you could think of this antigen as the face. The same exact way that your friends and family and colleagues and classmates look at your face and they recognize you as self or stranger. So the cell has a face, and that face is the antigen. So basically, your immune system will look at the face and will tell, okay, this cell is self, I'm not going to attack it. Or this cell is foreign, non-self, and I will attack it. Even if your phone, when you look at your phone, if the phone recognizes your face, your phone will open. If it doesn't recognize your face, your phone will not open. So the same way that we as humans, we have different faces, every single cell has different antigens. So in this case, our body is the cell and our face is the antigen. So our face is located on the surface of the cell, antigen, and the body is the cell itself. So different cells have different antigens. Now, your friends, your family members will not attack you, will not basically act strange against you because they recognize your face. They will welcome you. They will welcome you to their houses. They will open their arms and nothing is going to happen. However, if a sneaky face, if somebody that's stranger walks into your parents' house, you are welcome. But if a stranger walks into your parents' house, your parents will start screaming and say like, hey, I don't recognize that face. You're not welcome to my house. You're not welcome. So what do your parents do? They will take appropriate actions and kind of prevent that sneaky person to get into their house and property. So the same exact way, your cells, your immune cells circulating in the body, anytime they recognize a cell that has an antigen that's not familiar to them, a face that's stranger, not familiar, your macrophages will stimulate your B cells, and your B cells will produce antibodies, and antibodies will go and bind to those antigens. So what is antibodies? Antibodies are proteins. Again, they're produced by plasma B cells. So plasma B cells produce antibodies. Each antibody has different shapes. So you see this shape over here compared to this shape. The shape over here and over here are different because each shape will recognize different proteins. So antibody will circulate in the plasma and they will look for the shape that they recognize and then bind to it. Let's assume that this is your cell, this one is your cell, and this one is your cell. This one is not your cell. This is a foreign cell. Either has been transplanted, or let's say that this is bacteria, or an infected cell, or red blood cell from another person that's different than yours. So your immune system, your macrophages will go and check. So the macrophages goes and checks those antigens and says like, I'm happy, I recognize that face, I'm not going to react against it. Checks this one, this cell, I'm happy, I recognize this face, I'm not going to react against it. Checks this face, I recognize this face, I'm happy. Checks this face and says like, something is wrong, I don't recognize this shape. So when it doesn't recognize the shape, now your macrophages very mad. So this macrophage now is not happy with this face, with this antigen. So what does the macrophages do? Macrophages will attack the cell, destroy the cell. At the same time, it will stimulate T helper cell. So I will tell T helper cell, hey, I need help. There's a stranger in the body. Could it be whatever? So there is a stranger. So what is going to happen? T helper cell will stimulate B cells. Now, what does the B cells do? B cells will produce antibodies. That's how basically it works. So you understand where the antibodies come from. So the exact way that is going to look at, so you see this shape over here. So now the antibodies are produced. So what the antibody is going to do? This antibodies have shape over here. And this shape was produced only against this antigen. So you see the surface of this cell has this antigen. Does this match this? This part does not match. So when you look at it, this does not match. So this antibody cannot bind to this cell. It's going to move to the next one. It will check this cell. Again, when you look at this antigen over here, it doesn't fit. It doesn't bind this one. Keeps moving to the next one. If you look at the antigen of this cell, the antigen does not properly fit. And it's going to keep moving. When it checks this one, this antigen perfectly matches the antibody. So what the antibody is going to do, antibody is going to bind to this antigen. So when it binds to the cell, so right over here, this antibody is bound to the antigen of the cell. Because this antibody has at least 2 binding sites, it could bind to 2 or more cells. Some antibodies have more than 2, they have 10. So the cell, so you see this antibody bound to this cell and this cell, and now bound to more cells. So all those cells are bound to the antibody. So antibody is kind of like binds to them and holds them together, clamps them together. And that clamping is called what? Agglutination. So what is agglutination in terms of the red blood cells? Is that when antibodies bind to red blood cells, imagine this was red blood cell from a stranger. A red blood cell that you don't recognize as self. And those red blood cells, you had antibodies against them. So antibodies bind to them and agglutinate them together. So this is showing that this is the antibodies. The green ones are the antibodies. And the antibodies are binding to the red blood cells and clamp them together. And that clamping of red blood cells are called agglutination. Now let's move to the red blood cells. So red blood cells have 2 types of antigens, antigen A and B. The antigen A and B determines whether your blood type A, B, AB, or O. If you have antigen A on the surface of the red blood cells, you're type A. If you have antigen B on the red surface of red blood cells, you're type B. If you have antigens A and B, then you're type AB. If you don't have antigen A or B, then you're type O. Then there is the third antigen, which is called antigen D. Antigen D determines the Rh factor, whether you're Rh positive or Rh negative. If red blood cells do have antigen D, then you're Rh positive. If there is no antigen D, there is Rh negative. Now, let's look at this, how it works. So here is the red blood cells. Assume this shape is antigen A, this shape is antigen B, and this shape is antigen D. What antigen is on the surface of this red blood cell? I see that this antigens that's located on the surface of the red blood cell is this one. So my conclusion is that this red blood cells have antigen A. Because it has antigen A and it doesn't have any other antigens, my conclusion is this is blood type A. Now, do you see any antigen D? I don't see any of this on the surface of this red blood cell. So what am I going to determine? That there is no antigen D. So my conclusion is that this blood type is Rh minus. What kind of antigens are recognized as self? So this body, the immune system will recognize any cell that has antigen A as self. Any cell that has antigen B or D, they will be recognized as non-self and it will attack it. So this body will have antibodies against B and D. And we're going to discuss this. So here is in the plasma. In the plasma, you have antibodies against B. So basically, those antibodies are against this antigen. Any cell that has this antigen, this person's antibody will attack it. Now, our conclusion for this one is that this is blood type A minus. So one of the things that you're going to realize, even though the antigen D is missing, there is no antibodies against D. Why? Because the plasma of Rh negative individual does not contain anti-Rh antibodies initially. Those antibodies are present only if that individual has been synthesized or previously exposed to Rh plus red blood cells. And that exposure can only occur either accidentally or during some kind of transfusion. So we can explain that in a second. Let's assume that this body was given blood type B. What is going to happen? So if blood type B is given to this individual, so those are the B antigens. So this is blood type B because it contains that type of antigen. So you see the surface of the blood contains those type of antigen. Now, because this individual has antibodies against B, so those are the antibodies against B, what is going to happen next? Antibodies will go and bind to the surface of those antigen and clamp, agglutinate these red blood cells together, and as it agglutinates, the red blood cells will lyse and die. And that's why if you're given wrong blood type, those blood cells will be attacked by your body. Now, what if you get A plus blood type? So you see this blood type over here has two antigens. It has antigen A right over here. So it has antigen A. So we determine that this is blood type A. But we see another antigen on the surface. We say antigen D2. So we determine that this blood type is A plus. If A plus goes to the individual, because this person has A minus, its immune system will not recognize this shape as self. So this is a stranger. Even though it recognizes this one, but it will attack this protein. Because both proteins are located on the surface of the same red blood cells, by attacking those proteins, the whole red blood cell will be destroyed. So remember, we said initially you don't have antibodies against D. But because of this exposure, now what is going to happen? Your immune systems will look, so your macrophages will look at this and say like, I don't recognize that. That's stranger. So macrophages will go and tell helper cell. And the helper cell will stimulate the B cells. And what do B cells going to do? B cells will produce antibodies. Now you have antibodies against D. So initially you didn't have, but after that exposure, now you have antibodies against D. And what those antibodies going to do? Antibodies going to go and bind to those cells and basically destroy them. Moving forward, so if you look at this type of cell, what kind of antigen we see on the surface? We see antigen B. So what type of blood is this? It's B. Do we see any antigen D in the surface? No. So it's B minus. What kind of antibodies initially this blood will have? It will only have antibodies against A. So it will only have antibodies against this antigen. Now after the exposure to antigen D, it will have antibodies against D too. What kind of blood is this? I see antigen A right over here. I see this shape. And I see antigen D on the surface. B, not D. So because it has antigen A and B, what type of blood it is? It's AB blood. Does it have any antigen D? No. So it's AB minus. Because D is missing, what kind of antibodies it will have against? It will have antibodies against D. But only after exposure, not initially. Do I see any antigen? I don't see any antigen. I don't see antigen A. I don't see antigen B. So that means this must be blood type O. Is it blood type O plus or minus? It's minus because I don't see antigen D. What kind of antibodies would it have? Initially, it will have only antibodies against A and B. But after exposure to D, it will also have antibodies against D. And when we look at the plus, so what we see over here, we see a blood type with antigen A, and we see a blood type with antigen, red blood cells with antigen D. So this red blood cells contain antigen A and antigen D. So what is our conclusion? That's A plus blood type. What is missing? B is missing. Because B is missing, this blood type will have antibodies against B in the plasma. Same thing with this one. So this one has B and D antigens. So our conclusion is that this is B plus blood type. What is missing? A. So it's going to have antibodies against A in the plasma. So it will attack any blood cell that contains A. What do we see here? We see A, we see B, and we see D. Because it has A, B, and D, we say that this blood type is AB plus. What is missing? There's nothing missing, so there's going to be no antibodies against A or B or D. And this one is only antibodies, antigens D, so it's O plus. And what is missing? A and B, so I will have antibodies against A and antibodies against B in the plasma. Drop of blood, you put it on a microscope slide right over here, and let's say I divided the slide into 3 portions. I put a drop of blood here, I put another drop right over here, and I put another drop right over here. I placed anti-A here, anti-B here, and anti-D here. And I see agglutination here. If it's agglutination, what does it mean? That means those blood cells have those antibodies. So I had those antigens, and because I had those antigens, my antibody was able to bind to it and clamp them together. So the two cells are clamped together only if it contains those antigens. Because it has this antigen, now my conclusion that this blood has antigen A on the surface. Does it have any antigen B? No. Why? Because I don't see any agglutination. If agglutination doesn't occur, that means like my antibodies were not able to find the antigen and bind it. So you see, this is the agglutination. Because these antibodies are against A, against B in this case, so you see they bound to the cell and agglutinated it. Because no agglutination occurred, that means there is no antigen B. And agglutination occurred, that means antigen D is also present. If this blood contains antigen A and D, my conclusion is that this is A plus blood type. What do you see right over here? Right over here, you see agglutination occurred right over here. Agglutination occurred here. No agglutination occurred here. So no agglutination here, yes agglutination in those two. So what is my conclusion? That this is B plus blood type. Now, if you look at this one, agglutination occurred in all three of them. So agglutination is here, agglutination occurred here, and agglutination occurred here. What type of blood it should be? It should be AB plus blood type. And if I keep going and look at the next one, so you see no agglutination occurred here, no agglutination occurred here, and no agglutination occurred here. So what type of blood is that? O minus. Because this blood doesn't have antigen D, it doesn't have antigen B, and it doesn't have antigen A. With this, we conclude our video. Thank you.